All right, in this video, we are going to talk about venous return. And notice that venous return is of course going to impact end diastolic volume because that impacts how much blood will end up in our ventricles. So venous return is the return of blood to heart via the veins. That's why it's called venous return. It is going to impact and control the end diastolic volume. Thus, it'll impact stroke volume and cardiac output. There's a few things that venous return is dependent on. First off, blood volume and venous pressure, we'll talk about. Venoconstriction, which can be caused by the sympathetic nervous system. How well the skeletal muscle is activating and pumping blood back through the venous system, and also the pressure drop of blood within veins during inhalation. So at any given time, you've seen this before, at any given time at rest, there is more blood in our systemic venous system than anywhere else. Here we show our pulmonary circulation, here's our heart, and then here would be our arterial circulation. So at rest, at any given time, more blood is in our veins, at our systemic veins, than any other part of our circulatory system. So we're going to talk about how fluid flows, which impacts blood pressure. And I think you've seen this picture before, where, we have different compartments of fluid within our body. First off, we have intracellular fluid, so the intracellular compartments, and this would be inside the cells. And then we have extracellular fluids, which means outside of the cells. Notice that intracellular fluid, just based off of the numbers here, it's about 2/3 of body fluid, 2/3 of our body water. Whereas extracellular fluid is about 1/3 of that, 1/3 of our body's water. And extracellular fluid is actually broken into two parts because we have our blood plasma, and then we also have our interstitial fluid. Interstitial fluid just means the fluid outside of the cells. And we're going to talk more about what that interstitial fluid is. But of those two fluids, most of it is in our interstitial fluid, whereas only 1/5 of all extracellular fluid is within our blood plasma. These can be regulated or changed via water intake. And so down here it shows the GI tract increasing interstitial fluid, but also it can possibly decrease interstitial fluid by, excretion or excretion of waste. So we're talking about the movement of fluid through our tissues. And if we were to focus really just at our capillaries, there's going to be movement of fluid, as you can see from these yellow arrows and these orange arrows that are going towards the capillaries. These are showing the net movement, and the yellow arrows are pointing out a type of movement out of the capillaries. This is called filtration because fluid is moving out. It's like they're being filtered out of the capillary. This is caused by what we call hydrostatic pressure. Hydrostatic pressure is the pressure of blood. It's the pressure against the capillary wall, And so it happens because of the blood flow. And as we know, blood pressure is pretty high in our arterial system. So even though our arterioles are getting smaller, capillaries are getting smaller, and because of the surface area, because there is more surface area, the blood pressure does decrease here as it moves further away from the heart. But even then, there is still blood pressure coming through here. And that blood pressure is going to push fluid out of the capillary. This is called filtration caused by hydrostatic pressure. We're going to talk a little bit more about this. And so this usually happens on the arterial end of the capillary, so closer to the arteriole. And then there is a antagonist pressure or antagonist movement. And those are shown by these orange arrows, and notice they're going towards the capillary. This is caused by colloid osmotic pressure. Colloid osmotic pressure has to do with the plasma proteins that we have inside our blood. And, we've spoken about how if we have a lot of molecules, because of osmosis, our water in our body is going to want to go from high concentration to low concentration. So out here, the concentration of water would be high. Because of all of those proteins, it would be low within our blood. And so we would absorb more water back into the capillaries. This is due to colloid osmotic pressure. The word colloid just has to do with protein. And so this is going to make water be absorbed back into the capillaries. And so water or fluid is going to push back into our capillaries here. And so if you think about the pressure of blood within the capillaries, it is going to equal the pressure going out versus the pressure going in. So the pressure going out versus the pressure going in. And notice if you look at this, calculation, there is colloid osmotic pressure inside and outside. And there is filtration pressure inside and outside. And so they're both going to have an effect on the net filtration and the net absorption. So these are called Starling Forces, and this is going to come up again because, we have capillaries everywhere and they impact how fluid moves through our capillaries. So there is the net filtration. So when we have a capillary and there's going to be pushing out of fluid by the hydrostatic pressure, or like the pressure of the blood pushing through the capillary. The net filtration would be the difference between the hydrostatic pressure of the blood plasma, so what I drew in blue, as well as the tissue fluid, so what I'm drawing in green. Because there is also going to be fluid out here, right? I can draw it in blue maybe. There's going to be fluid out here that also pushes back against the capillary. It's not going to do it as much, and so maybe I could draw these arrows a little smaller. It's not going to do it as much as the hydrostatic pressure of the blood plasma. So net, it would be a greater push out. There would be more filtration out. However, there is a second type of force to consider here, and that would be colloid osmotic pressure. There are proteins within our blood, and because of that, it's going to want to favor fluid coming into our capillaries. This is called colloid osmotic pressure, and this would be the colloid osmotic pressure of the blood plasma. However, just as there was hydrostatic pressure of the tissue fluid, there's also colloid osmotic pressure of the tissue fluid, and that is also going to cause some movement of fluid out of the capillary, just like that. There is less protein in the interstitial fluid than there is in the blood plasma. And so again, absorption here would favor, coming in towards the blood plasma. So these Starling forces, the fact that there is hydrostatic pressure on the blood plasma side, the tissue fluid side, as well as colloid osmotic pressure on the blood plasma side and the tissue fluid side, fluid is going to leave capillaries at the arterial end because of this hydrostatic pressure, but then it returns back into the capillaries at the venial end because of the colloid osmotic pressure. So we're going to talk about the lymphatic system in just a second here because it's super relevant to this, but maybe you've heard of tissue edema. Edema is tissue swelling, and that's caused by accumulation of tissue fluid. And this can be caused because of these Starling forces when there is too much filtration and not enough reabsorption of the tissue fluid. This can be caused by a few things. High blood pressure, right? And that means that net filtration pressure will be really high. Venous obstruction, again, that would decrease the amount of absorption back into the capillaries. Leakage of plasma proteins into the extracellular fluid. If there were plasma proteins in the extracellular fluid, if there were more out here, then the colloid osmotic pressure is going to favor going out. Myxedema, which is similar to the leakage of plasma proteins, it's going to cause more proteins in the extracellular fluid. Low plasma protein levels, which similar to these two, is going to favor a higher colloid osmotic pressure into the interstitial fluid, or obstruction of lymphatic drainage. And I'll show you an image of this. If you can recall from, I don't know, a week or two ago, we spoke about the lymphatic system and how lymph is basically interstitial fluid, right? Plasma that is pushed out of our capillaries and absorbed via our lymphatic vessels, just like shown. Maybe I'll do this in green because lymph is usually shown in green in textbooks. This is done by these Starling forces. And the only thing that's different here is that our lymphatic vessels, our lymphatic capillaries here, are picking up that excess interstitial fluid. And if you can remember from that lecture a couple weeks ago, lymphatic vessels are going to pick it up and it's going to take it to those checkpoints, which are our lymph nodes, to make sure that there are no foreign invaders like bacteria or viruses, because they'll have to go through these checkpoints, which are basically B cells and T cell accumulations, and it'll cleanse the plasma and then return the plasma back to the venous system of the blood. So our lymph is doing us a favor because it's picking up this excess tissue fluid. In this image, it shows excess fluid being picked up by the lymphatic vessels, being pushed through our lymphatic capillaries, taken to a lymph node where they're being cleaned, and then returned to the venous system. So we just said, though, how obstruction of lymphatic drainage can cause tissue edema. And this is a good example, clinical example of this. This is called elephantiasis, and it's caused by a worm infection, filaria worm infection. And what it does is it blocks lymphatic drainage. So the larvae of these worms block lymphatic drainage, and that is going to block off the capacity that your lymphatic vessels have to remove that interstitial fluid. And so what it does is it causes tissue edema. It's going to cause tremendous enlargement of specific areas, perhaps your genitalia, because there is no drainage of your interstitial fluid through these lymphatic vessels. To treat this, temporarily, you could release maybe release some of the fluid here and allow it to drain, but this would only be a temporary fix. So now we're going to talk about 2 or 3 different hormones that are going to impact blood volume. These are going to impact blood volume. The first one we have here is called ADH. And if you can remember, ADH is produced by our hypothalamus and it is released by the posterior pituitary. It is produced when you're dehydrated, when your blood volume is low. And so when blood volume is low, that makes your blood osmolality high. Your blood osmolality is high. And that is what causes osmoreceptors in the hypothalamus to produce this hormone. It will be released by the posterior pituitary, and what it does is it causes water retention by the kidneys. This is like when you're sitting in lab on Saturdays for 6 hours and all your water bottles are outside and you can't drink water when you want to, and so you're feeling a little thirsty, but you're in lab, so you're not drinking as you normally would be. And so what happens is your hypothalamus will release ADH, and what it does is it will cause water retention in the kidneys, and it does this by reducing the number of aquaporins. And so you will not urinate. You urinate less when you are not drinking water. And that is thanks to ADH. And so it's really important, and it's a good thing that we do not urinate as much as when we're not drinking as much fluid, so that our blood volume can stay relatively high and reduce our blood osmolality. Coffee and alcohol impact ADH. And so if coffee makes you urinate, alcohol makes you urinate. And so coffee and alcohol block the action of ADH. And so this is why you're dehydrated after a night of drinking. It does not retain water in the kidneys. Aldosterone is a mineral corticoid, and it is produced by our adrenal gland. And it also is going to impact blood volume. It does it in a different way. It's called a mineral corticoid, and so it's going to impact the minerals. And it does this by retaining salt. It makes your body retain salt. And in effect, if you retain salt just based off of osmosis, you'll retain more water. And so in that way, it'll keep your blood volume high. It'll keep your blood pressure high. And so this will, act as a negative feedback loop. Aldosterone is kind of known as like the hormone to control it all, because it really does have big effects on blood pressure and blood volume. So this would be secreted when blood volume is low, maybe when you're dehydrated, or when your blood pressure is low. So we're going to talk about this system again, but we're just introducing it right now. We will talk about this again when we get to our renal system. But aldosterone is kind of known as the hormone that controls it all, and it feeds into this bigger network of a system. And so I'm just introducing it right now, and I know that this pathway looks like a lot, but let's just break it down a little bit. Before we talk about that, I want to show you what your nephron looks like, and hopefully this is review from human anatomy. So this is showing blood flow through a nephron, and this is where filtration occurs, right? And by the way, Starling Forces occur here too, and we'll talk about that when we get to the renal system. But your kidneys do really important work, right? And it is fully dependent on blood pressure. It is totally dependent on blood pressure. But as you know, our blood pressure changes all the time. It changes when you wake up, it changes when you go to sleep, it changes when you're asleep, it changes when you exercise, it changes when you're stressed out. And so our kidneys do really important work. We wouldn't want our kidney function, our nephron function, to change whenever our blood pressure changes, because our blood pressure changes all the time. And so we'll get into more detail later, but there is a complex here called the juxtaglomerular apparatus that is going to regulate how fast or slow blood pressure is coming through these blood vessels. And the way it does that is by producing an enzyme called renin. And renin is an enzyme that is going to go into the blood, and your liver produces something called angiotensinogen. So this is just hanging out in the blood, all the time. And so whenever our kidneys, or whenever the juxtaglomerular apparatus produces renin, it is going to convert this angiotensinogen into this angiotensin 1. And then there is something in our lungs called ACE. ACE stands for angiotensin converting enzyme. It lives in our lungs, and that's because all of our blood flows to our lungs. 100% of our blood has to go get oxygenated in the lungs, and that's why the ACE enzyme lives there. So whenever there is angiotensin 1 hanging out in the blood, it will come into contact with this ACE, and then it becomes angiotensin 2. What angiotensin 2 does is two things. First off, it's going to act on the adrenal cortex, and that'll stimulate the adrenal cortex to make aldosterone. That is going to cause salt retention, and if it causes salt retention, it's going to cause water retention too, and this is going to increase blood volume. That's one of the ways angiotensin works. The second way it works is more of a direct effect, and it is going to directly affect our arterioles by vasoconstricting them. And right away, if you vasoconstrict your arterioles, that is going to increase your blood pressure. And so, this is kind of like a really big pathway, and this is going to come up a lot, so if it's confusing now, it's okay. But this is a really big way in which a lot of different parts of the body, the liver, the kidneys, the lungs, the blood, the adrenal gland, all control blood volume and blood pressure. The last hormone we have here is called atrial natriuretic peptide, ANP, and it's going to be antagonist to aldosterone. It's going to do the opposite of aldosterone. And so increased blood volume, which means there's going to be increased venous return, which means there's going to be increased stretching of the left atrium. This is going to cause release of ANP. And what that does is it does the opposite, it's antagonist to aldosterone. It's going to increase salt excretion, and so if it increases salt excretion, water is going to follow, and it increases water excretion. This is going to increase our urine volume, but it's going to decrease our blood volume. So again, this is a negative feedback loop. When there is increased blood volume and increased venous return, and thus increased stretching of the left atrium, the vagus nerve is going to signal to the brain to decrease the release of ADH. So ADH secretion is going to decrease, water reabsorption is going to decrease, that'll increase urine doing the opposite here, and then decrease blood volume. Talk about factors affecting blood flow. This is showing pressure, and these are showing the different blood vessels, right? And closest to the heart, we have the left ventricle, part of the heart. Notice that the pressure is really high. It goes to low though when the ventricle is emptied. And then we have our large arteries, and then they move away from that, our smart arteries. But notice that blood pressure is high. High blood pressure from the heart, still pretty high here. Maybe I could do 2 arrows over here, 1 arrow over here. And then from the capillaries to the veins, very low blood pressure. And so I think I've mentioned this before. As we move away from our heart, blood pressure decreases. And blood pressure decreases because surface area as we move along increases, increasing surface area as we move along. And then here we have the Starling Forces pushing blood out. And so that's also going to decrease our blood pressure. So blood flows from highest pressure, higher pressure, to lower pressure. Higher pressure in the arteries to lower pressure in the veins in our systemic circuit. We can define blood flow as delta P over R. So delta P is the pressure difference between the two ends of the vascular system. So like between the arterial end and the venous end. And R is the resistance or the opposition to flow. So if delta P increases, blood flow will increase. And if R increases, blood flow will decrease. Just based off of this formula right here. But what it's showing is the changes in pressure. And I guess the story is if the pressure here at the end equaled the pressure at the beginning, there wouldn't be any movement. So if this was 100, and this was 100 too, that means like there wouldn't be any flow of blood. So, whether there is blood flow is dependent on the change of blood pressure. And so here at the beginning of our arterial system, it's about 100 millimeters mercury. And at the end, right at the vena cava, it's 0 millimeters mercury. And so our delta P here is 100 millimeters mercury. And this is what allows for the pathway of blood to be able to be pushed through our arterial system, our capillary system, our venous system of the systemic circulation. So blood flow equals delta P over resistance. And so what this is showing you is 2 different arteries and how the size of the arterioles are going to change resistance and blood flow. The bigger the tube, the less resistance, because resistance is proportional, this is the sign for proportional, to the length of the vessel, the viscosity of blood, and then inversely proportional to the radius of the vessel. So what that means is if this number is bigger, that means there's less resistance. If this number is smaller, that means there's going to be more resistance. And the longer the length of a blood vessel, the more resistance. The more viscous blood is, the more resistance. And so let's look at these 2 examples here. This shows one artery that breaks into 2 smaller arterioles. And you can see that the radius is equal to the same, and the resistance and blood flow are the same for both of those vessels, as expected because the radius is the same. Let's look at this bottom example down here. So this example is showing 2 millimeters. This one is showing a 0.5 millimeter. So that would be a fourth of what this is, a fourth of this top one. Notice though the difference between the resistance and the blood flow. The blood flow here is 16 times greater than the blood flow in these top examples. And the blood flow here is 1/16 of the blood flow at the top. The resistance here is 16 times the resistance of the top. And then the resistance of the bigger vessel is 1/16 of the resistance here. And so the size of the vessel matters. And the smaller the vessels are, the more resistance there will be, the less blood flow. So another way to say this, or just written down, blood flow is directly related to the pressure difference at the two ends of the vessels. Remember we showed you Remember we showed you arterial pressure and then venous pressure. So delta P would be 100. And that's what allows for blood to flow through. But it is inversely related to the resistance. The more resistance, the less blood flow. The less resistance, the greater the blood flow. Resistance is going to be directly proportional to the length of the vessel and the viscosity of blood, but inversely proportional to the radius. And that means the bigger the radius, the less the resistance. So with this law called Pucel's law, vessel length and the blood viscosity, blood viscosity does not change usually, and vessel length does not change either. It's like a closed system. So what really matters here are the arterial pressure as well as the radius of the blood vessel. Those are the most important factors that are impacting blood flow. And so if you think of vessel radius, this is where vasoconstriction and vasodilation occur. Vasoconstriction is going to provide the biggest resistance to blood flow, and that is going to directly impact the flow of blood to and from organs. There is paracrine regulation of blood flow. If you can remember what paracrine was, it's kind of like a local signal. So an example of this would be the endothelium producing molecules that are going to act on the smooth muscle layer of the vessels. And some examples of that, one big one is nitric oxide. Nitric oxide can be produced by the endothelium of our blood vessels, and that is going to cause vasodilation. Some other examples are bradykinin, prostacyclin. Those are all vasodilators. They're going to increase the radius of our blood vessel. Endothelin 1 is an example of a vasoconstrictor. It'll decrease the radius, which will increase the resistance. So this is an example of autoregulation. Auto means self, right? So this organ, the blood vessel itself, is regulating itself. So autoregulation is the ability of an organ to intrinsically, so it comes from itself, modify the degree of constriction or dilation of its arterioles, and thus regulate the rate of blood flow. Another example of an organ that does this is your kidneys. And so similarly, when we were talking about aldosterone and how it turns that on, it can also just change the smooth muscle in the vessels itself. So the juxtaglomerular apparatus can change the arterioles here too. So this can happen in this example, this would be a myogenic mechanism, but in other examples, it could be myogenic or a metabolic mechanism. So an example of myogenic regulation is with vasodilation and vasoconstriction of your blood vessels. And a good example of this would be in the brain. So when there is decreased blood pressure to the brain, the smooth muscle of the blood vessels themselves are going to release a paracrine response that will increase blood flow. And contrastly, if there's increased blood pressure, it'll send a response that will decrease blood flow. This would be considered myogenic because it's the smooth muscle within the blood vessel walls itself. An example of metabolic regulation would be the changes in the tissues, the changes in the chemical environment that are going to cause this change of blood flow. So if there was low oxygen or high carbon dioxide, that can change the vasodilation or vasoconstriction within that area.